My name is Paulina Dragan, and the poster I'm presenting is titled Insights into the Orexin Receptor Type 2 Mediated Activation of G-alpha-Q Provided by Molecular Dynamics Simulations. Orexins are hypothalamic neuropeptides known for their involvement in the regulation of the sleep-wakefulness cycle. They activate two receptors, OX1R and OX2R, which couple primarily with G-alpha-Q. Orexin receptors are abnormally expressed in different cancers, for example OX1R in glioblastoma and colon cancers, and OX2R in endometrial carcinomas and cortical adenomas. This suggests that the orexin system could be targeted in cancer treatment. Orexins can induce apoptosis through an SRC kinase signaling pathway, an effect that has also unexpectedly been observed for orexin receptor antagonists such as almorexant, lemborexant, and daridorexant. In order to determine the theoretical basis of this observation, we performed molecular dynamic simulations for OX1R and OX2R bound to mini-GSQI, or the full-length G-alpha, as well as the non-peptide agonist compound 1, or lemborexant. Our models were prepared using a combination of Modeler and Rosetta. The templates we used were 7L1V for the OX2R receptor and the mini-G protein, and 7SQ2 for the full-length G-alpha-Q. These models were then pre-processed with Maestro, and the simulation systems were prepared using CharmGUI. The simulations ran for 2 microseconds and were performed in NAMD. As a result of the simulations, we found that both the agonist compound 1 and antagonist lemborexant were stable in the orthosteric binding site of the active state OX2R receptor throughout the 2 microsecond simulations. We performed similar, albeit shorter simulations for the OX1R receptor and obtained similar results. In addition, we observed that ECL2 moved to cover the extracellular side of the receptor during the 2 microsecond simulations. This close conformation of ECL2 was attributed to the formation of polar contacts between certain residues. The closing of ECL2 was observed for both lemborexant and compound 1. Changes in the conformations of the residues FGH508 and DGHGO2, suggested by Hem et al. To be microswitches, were observed, and this confirmed the beginnings of G-alpha-Q activation. We also observed the beginnings of GDP dissociation, and the residues near the binding site rearranged to create an exchange tunnel near the phosphate site. The global conformational changes of the simulation systems were compared to those of the 6-CMO PDB structure, representing a rhodopsin G-alpha-I complex, where the alpha helical domain of G-alpha-I has separated from the Ras-like domain. In the simulations, the helical domain began to move in the same direction as in the 6-CMO structure via a hinge consisting of helix HF. Prior to this movement, the 3 macroswitches, especially switch 1, also started moving in the same direction as in 6-CMO. Interestingly, the separation of the helical domain from the Ras-like domain appeared to happen faster for the lemborexant systems. To conclude, the molecular dynamics simulations confirmed that there are conditions under which lemborexant displays agonist-like behavior and stabilizes the active state OX2R receptor. In the experimental study, this effect was observed in the HEC293T cell line and HT29 and HCT116 colon cancer cell lines. In addition, G-alpha-Q activation occurred more quickly in the lemborexant systems as compared to systems with compound 1. The faster GDP to GTP exchange observed for lemborexant might further evoke the G-beta-gamma mediated activation of SRC kinases. Furthermore, a series of conformational changes that occur during the OX2R agonist-mediated activation of G-alpha-Q was proposed. First, we observed a change in the switch FGH508. Then, a breaking of the interactions between the GDP molecule and RGHFS202 located on macroswitch 1. Instead, this arginine then formed interactions with EGS1H104, which was located on the P-loop. These interactions were later broken, as were the interactions between EGS4H3.14, located on switch 3, and 2 residues located on switch 1. EGS4H3.14 then went on to interact with RGHFS202, And around this point, we observed the formation of the GDP to GTP exchange tunnel. Then the interactions between RGHFS 2.02 and EGS4H 3.14 were broken, as were the interactions between the GDP molecule and DGHGO2. All of this leads to the separation of the alpha helical domain of G alpha from the Raslake domain and subsequently GDP release.